In this lesson, we are going to talk about mutations that may occur in DNA and their potential effects on the organism. So first of all, a genetic mutation is any change in the DNA sequence caused by various mechanisms, which could be exposure to synthetic chemicals or to radiation. It can also be caused by incorrect replication and just random mutations that may occur in cell division. Okay, your textbook likes to talk about the effects of smoking, as it very well should. And there's a specific locus on chromosome 15 that if you are a smoker and you develop a mutation at this point, the locus is 15q24, so that's the location of the gene, you end up with a 30 to 80% increased chance of developing lung cancer. So a susceptibility locus is a region on a given chromosome, so chromosome 15 in this case, where mutations that affect one or more genes are more likely to be present, and this is based on statistical evidence. So we're going to talk about different types of mutations. So the first umbrella term is a point mutation. So this is any change in a single nucleotide. So the first one is a substitution. So this is a simple replacement of one nucleotide in a DNA sequence by another nucleotide. So the example shown here is thymine is being replaced by a cytosine. The next example is a insertion, and these are called small-scale mutations because they are literally happening typically with one nucleotide, so they call it a small scale. It can still have large effects, though. So this is the addition of a base pair or even a larger coding region, and we'll talk about the larger coding regions a little later in the lesson. So in this example, we have our normal sequence, and we see what the mRNA sequence is and then what the amino acid sequence should be. But we have an insertion of a cytosine. So instead of being CTT, this C got replaced, and that's going to move everything down. So instead of being CTT coding for glutamic acid, it is now CCT, which will code for glycine. And then that moved this T over, so now it's TCT, which codes for arginine instead of another glutamic acid. So everything downstream of that insertion typically gets changed, unless you're lucky and you might get one or a couple amino acids that are the same. So similarly, you get a deletion where a nucleotide is removed. So in this example, the adenine is being removed. So the adenine should be here, but it's removed. So that moves everything. Let's go the right up. Trying to go the right direction for you guys. Moves everything up. So instead of ATG coding for tyrosine, that A is removed, so it would be TG, and the C is now part of that codon. So you get threonine, and everything else is shifted. So you would get lysine instead of glutamic acid and arginine instead of the second glutamic acid. So that is the result of a single nucleotide deletion. An inversion is where you have breaks in the DNA, and the part that broke just flips around and then gets ligated back in, just backwards. And so in this case, we had the break here and after the adenine, and then they flipped around. So that's an inversion. So single nucleotide polymorphisms are just an interesting side effect of a point mutation. So somewhere in non-coding DNA, so in the introns, for example, there might be just a single nucleotide difference between individuals in a population. So one individual might have an A, another a G, another a T in the same spot, and it doesn't have an effect on the organism. So it would be in a non-coding region. These are useful in forensics and paternity tests, and we're going to have some fun with that a little bit later in this unit. So there are different effects that mutations will have on the organism, or essentially in the creation of the polypeptide. So a missense mutation is one that changes a single amino acid in the coding sequence. So our example is here. So the T got changed to an A, and originally there should be a cysteine, but instead it codes for a serine. Everything else is the same. So there is a good chance that maybe this won't have an effect, or it won't have too much of an effect on the functioning of that protein. A nonsense mutation results in a premature stop codon. So in any codon where you get a stop sign instead, or a stop codon, I call it a stop sign sometimes, instead of an amino acid, then you are truncating, you are shortening that polypeptide. This is an extreme case where we have our start codon and immediately we have our stop codon. This obviously would have, in most cases, a serious effect on the functioning of that protein. Unless maybe you are, it's just maybe at the very, very end, it may shorten it a little bit. So it really depends on the location of that stop codon. Okay. You might also get a silent mutation, and this is one of the wonderful things about the wobble effect. So remember, in a lot of scenarios, that third nucleotide in a codon could change, and you still get the same amino acid. So that would result in, that would be called a silent mutation. It's having no effect whatsoever on the production of that polypeptide. Okay, we also have a frame shift. So this is when we have a shift in the reading frame, resulting in multiple missense or nonsense effects. So this is a scenario where, for example, if you have a deletion or insertion, and then everything in that reading frame gets shifted up or down, and then all of the amino acids or most of the amino acids change. So here's our example where we have our DNA sequence and we have our RNA sequence, and we are coding for tyrosine, leucine, arginine, arginine, and alanine. However, if that G is removed, everything else gets shifted up. Like so. So now we still have our methionine, but everything else is different. So there's our RNA strand, and now we're getting tyrosine, phenylalanine, valine, and then, if something is following this, you would get histidine or glutamine. So everything is shifted from downstream. Large-scale mutations can involve multiple nucleotides, an entire genome, or whole regions of chromosomes. And so there's different types of large-scale mutations. So we're just dealing on a bigger region of DNA being affected. So one is called amplification. So this is where we have a gene or a group of genes is copied to multiple regions of the chromosome. This can compound the effects of the gene. So it takes a little bit of thinking to think about that. So if you have an entire gene with its promoter in multiple spots in that cell, and they're all getting turned on, they're all working, then you can almost imagine it's being like times 3 the production. Or if you have a single double, let's use the word double. So a gene is copied and it's somewhere else and it has its promoter and it's all intact, then you're getting twice as much production of something that potentially you don't need. What's really interesting is you might have some mutations that occur in one of those genes, and you could actually develop new functions. And so again, from last year, thinking about how we evolve, and it's through mutations and natural selection. So if a mutation benefits the organism, then they will reproduce more, and those mutations will grow in population. Okay, there are deletions, and these may negatively affect the organism. So dystrophin is a protein that is an important component of skeletal muscle. Deletion of part of the gene results in Duchenne muscular dystrophy. Individuals usually die in their 20s due to weak respiratory muscle. A small part of the gene is deleted, so not the whole gene, but just a small part of the gene. So Duchenne muscular dystrophy is typically the most severe, and it has the largest deletion. Becker has a smaller deletion of that gene, and they have weak muscles, but they otherwise can have a normal life. You can have large-scale mutation with a translocation. So you can have an entire gene or a group of genes moved from one chromosome to another. And so we see here we have this blue section of this chromosome number 4 gets switched with this green section of chromosome 20. So you can have a translocation between two non-homologous. So non-homologous would be 4 and 20. They're not both chromosome 4, both chromosome 20, one from mom, one from dad. And this occurs when portions of each chromosome breaks off and changes places. And you would learn more about that in university. So there's a lot that goes on in that we don't start to talk about in grade 12. Okay, there's really cool things called transposable elements. They're also called transposons and accurately also called jumping genes. So these are sequences of DNA that move freely around the genome. If they are inserted near an existing gene, they can enhance that gene, or they could disrupt that gene, or they can somehow modify the expression of that gene. So it really depends on the gene itself, the transposon, and the location of where it inserts. So a lot of different things could happen. Essentially, it could be causing an inversion. It could be causing a large insertion, or it could be somehow causing RNA polymerase to bind to it more likely, or it could be truncating it. So lots of different things could be happening depending on those factors. You can also get an inversion, so a portion of DNA, and again, we're talking about large scale here, so not just like a nucleotide or two, but we're talking about a whole gene or two can actually reverse in its direction in the genome. If the whole gene's reversed, there might actually be no effect. However, if an inversion happens in the middle of a gene, then the gene may be compromised. Trinucleotide repeats are common in our genome of healthy cells, healthy individuals. But what happens is you might get an expansion of those nucleotides. So tri meaning 3, so you have 3 nucleotides that repeat. So in the Huntington's case, there is a repeat of CAG. So again, it's normal to have a certain amount of repeats in a healthy individual, but once they become too high, you can be predisposed to having a certain disease. So in Huntington's shown here, 10 to 26 repeats are normal, but over 37, you are looking at having that Huntington's disease. There's also myotonic muscular dystrophy, which runs in my family. And this is caused by a repeat expansion in one of 2 genes, so DMPK or Z and F9. So what causes genetic mutations? We talked a little bit about the beginning. So some are spontaneous and they arise from inaccurate DNA replication. So mistakes happen, and sometimes, most of the time, they get caught by polymerase 3 or polymerase 2, but sometimes they just get missed. They can be induced by environmental agents that directly alter DNA in a cell. I'm going to talk about a couple of those for our last couple slides. Mutagens, so a mutagen is any kind of chemical that can enter the nucleus and can directly alter the genome. So what are some of these chemical mutagens? So again, chemicals that enter the nucleus and can alter the structure of DNA. We have carbon monoxide, alcohol, and tobacco smoke are all known to lead to cancer. There is nitrous acid, can modify individual nucleotides so that nucleotides resemble other base pairs. So they're not even switching nucleotides, they're just changing the nucleotide itself. Because if you look at them, purines kind of look the same, pyridines look the same, and so they can actually go in and modify those. This confuses replication machinery and creates inaccurate copying. Benzene binds individual nucleotides and can be added to replicating DNA. So it's not even a nucleotide, but it can actually be added into the DNA. So this is going to change the shape and the effects of the DNA. Ethidium bromide is similar to a nitrogenous base in structure, and it can insert itself between the strands of the double helix and again change the shape of the DNA. It can also lead to replication mismatches. So radiation, we try to avoid, we know this. And this is sort of some background information about why. So we have electromagnetic radiation can cause mutations. So we have ultraviolet B radiation coming from the sun, can cause bonds to form between adjacent, so side by side, nucleotides. So this is going to cause a kink in the backbone of the DNA. This can complicate replication and transcription. This is often known to lead to certain types of skin cancer. There's also ionizing radiation, so which strips molecules of electrons. Here we go again. Strips molecules of electrons and will break bonds within the DNA. So we end up with a rearrangement or a deletion of large portions of chromosomes because of this. X-rays, for example, will cause certain types of cancers and tumors. So that's why when you're getting X-rays, you do that minimally, and if so, that's why you put on that big lead on whatever is not being X-rayed. Gamma rays in radioactive decay of nuclear material, for example, Chernobyl. A lot of children were born with birth defects. I have the friend who was raised in Poland after Chernobyl, and she was on the border of Poland and Ukraine, and her whole family is riddled with cancer. There's the 2011 tsunami that induced the nuclear meltdown in Japan, and there's known to be some medical or health defects or repercussions after that. Okay, so good or bad? Mutations, hopefully you learned last year a little bit how they are good. They create variations and diversity, which is awesome in itself. So if we were all the same, we'd be boring. Mutations that negatively affect an individual will usually be selected out by natural selection. An advantageous mutation will allow the individual greater survival benefits and be passed on as they will procreate more. Most mutations are neutral, yay, and they have no effect on the organism. So about 90% of the DNA is non-coding. Prokaryotes are less likely because most of their DNA is coding. Okay, and some common mutations that are interesting to know about. So lactose tolerance in humans. If you can drink milk, you don't need to, but if you can, that's due to a mutation. Sickle cell anemia, the mutation that causes it can actually, so having sickle cell anemia can actually protect against malaria. What's nice, or more beneficial, we'll say, is if you only have one allele for sickle cell anemia, and then you have the normal cell shape, then you would have half healthy cell red blood cells and half sickle shaped. So you don't have the sickle cell anemia effects too badly, and you still have the malaria protection. A mutation in the gene filaggrin is an increased risk, or leads to an increased risk in peanut allergy. So 1 in 5 peanut allergy sufferers have this mutation. So that's everything that we are going to talk about with mutations. So there's a lot of definitions in this lesson. You would be expected to know the different types of mutations and their effects, and we will talk more about this tomorrow. We'll review it, and we have a little activity to go with that.